Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Okay, so imagine this. What if a protein, something totally essential, for building the very blood that keeps us alive, what if that same protein was actually secretly helping cancer hide from our immune system?
It sounds almost like science fiction, doesn't it?
It really does.
Yeah.
But that's the surprising role researchers have just uncovered for erythropoietin, or, well, EPO people sometimes call it, but erythropoietin.
Yeah. And what's truly fascinating here is that for nearly 4 decades, we've mostly just thought of erythropoietin's main job stimulating red blood cell production. That's what it's known for.
Right, the basics.
Exactly. But this new research, it was just published in Science, it reveals this completely unexpected side. It can actually dampen the immune response, specifically inside cancerous tumors.
Wow.
It really forces us to rethink why some cancers are just so incredibly hard for our own bodies to fight off.
Okay, yeah, we definitely need to unpack this. It sounds major. So this deep dive, we're going to focus on this new study, and there's also a related report from Stanford University Medical Center we'll draw on.
Good plan.
And our goal, really, is to get our heads around how blocking the signals from this protein, erythropoietin, how that could potentially turn these cold liver tumors in mice.
The ones the immune system ignores.
Exactly, the ignored ones.
Yeah.
Into hot tumors. The kind of the immune system can actually see and attack.
Mm.
It really feels like this could open up a whole new avenue for cancer immunotherapy, maybe especially for those cancers that just, haven't responded well so far.
Absolutely. I mean, that's a huge challenge in cancer treatment, isn't it? While some cancers respond brilliantly to immunotherapies, you hear about the successes, especially with treatments targeting the programmed death-1 molecule on T cells.
Yeah, the checkpoint inhibitors.
Right. But unfortunately, many others just don't. We're talking about a lot of liver cancers, pancreas, colon, breast, prostate cancers.
The common ones often fall into this category.
Very often. And they get called cold tumors for precisely that reason. They just lack that significant infiltration, that presence of the crucial cancer-fighting T cells.
So walk us through the connection then. How does this protein, erythropoietin, which we associate with blood cells, suddenly get involved with these cold tumors?
Well, the researchers noticed something really key. These cold liver tumors in the mouse models, they consistently had much higher levels of erythropoietin compared to the hot tumors, the ones immune cells were getting into.
Okay, higher levels. Why?
The most likely culprit seems to be the low oxygen environment inside those cold tumors. It's a condition called hypoxia.
Right, lack of oxygen.
Exactly. And this hypoxia seems to trigger the cancer cells themselves to ramp up production of erythropoietin. The thinking is maybe they're trying to stimulate more red blood cell formation to get more oxygen delivered to the tumor.
Huh. So the tumor's almost trying to, fix its own supply problem.
Precisely.
But in doing so, it accidentally creates this other problem with the immune system.
That's wild. But hang on, haven't we heard before that erythropoietin and cancer wasn't always a good combination? You're absolutely right. That connection isn't entirely new. Actually, more than a decade ago, some earlier studies raised concerns.
All right.
They found that when erythropoietin was given to cancer patients, often to treat anemia alongside their cancer, sometimes their tumors seemed to grow faster.
Oh, wow. Okay.
Yeah, it was serious enough that the Food and Drug Administration actually issued a pretty strong warning, a black box warning back in 2007, basically advising against using erythropoietin in cancer patients if possible.
Right.
And there were also other studies finding correlations, suggesting that higher natural levels of erythropoietin and its receptor within tumors seem linked to a worse prognosis for patients.
So there was already some suspicion around erythropoietin maybe fueling cancer growth.
Yeah.
But this new finding is different, isn't it? It's about the immune connection specifically.
Exactly. That's the novel part. The link to cancer immunity. That's the real breakthrough here.
So how did the researchers actually demonstrate that link? What did they do in the lab?
Well, the lead researcher on this study, David Kungton Hsu, used some really quite sophisticated genome editing techniques. They created various mouse models of liver cancer.
Okay.
And these weren't just any models. They were specifically designed to mimic the kinds of genetic mutations, the histology, the way the tissue looks under a microscope, and even how they responded to therapies, mimicking what's seen in different subtypes of human liver cancer.
So really trying to replicate the human disease in mice.
as closely as possible.
Yeah.
And they induced the tumors either by injecting specific DNA sequences known to cause cancer or by actually implanting liver cancer cells directly into the mice.
Okay, and then they started testing immunotherapies on these different models.
That was the next step, yes. They specifically looked at an immunotherapy targeting that programmed death 1 molecule on T cells we mentioned earlier.
Remind us how that works again.
Sure. So programmed death 1 normally acts like a brake on T cells, stopping them from going overboard and attacking healthy cells.
Go.
But cancer cells are sneaky. They can display molecules on their surface that basically push that brake pedal on the T cells, telling them, nope, don't attack me. This effectively shuts down the immune response right where the tumor is.
Right, the cancer puts up a do not disturb sign for the T cells.
Exactly. So drugs that block that interaction, the anti-programmed death 1 therapies, they can essentially release the brakes, allowing the T cells to recognize and attack the cancer. And they work really well in some cancers.
Got it. And then there are different mouse models. Do they see different responses?
They absolutely did. They found that certain combinations of those genetic mutations they engineered led to these cold tumors. The immune system just wasn't getting in there.
Mhm.
And crucially, these cold tumors didn't shrink when treated with the anti-programmed death 1 therapy. Why? Because there were hardly any T cells inside the tumor to be unleashed in the first place.
Makes sense.
But other genetic combinations resulted in hot tumors packed with T cells. And those tumors, they responded well to the anti-programmed death 1 treatment. They shrank.
Okay, so this sets the stage perfectly for the erythropoietin connection. What happened when they started messing with the erythropoietin levels in these tumors?
This was the really crucial experiment. They modified the tumor cells so they couldn't produce erythropoietin as well or at all.
Okay.
And what they found was striking. When they stopped the cold tumors from making erythropoietin, those tumors actually turned hot. Suddenly, immune cells, T cells, started flooding in.
Wow, just by blocking the erythropoietin.
By blocking its production by the tumor cells. And they did the reverse experiment, too. They took the hot tumors, the ones the immune system was successfully attacking,
Right.
and they engineered those cells to produce extra erythropoietin.
And what happened?
Those hot tumors became cold, they became resistant to the immune attack, and started growing again, even with the immunotherapy.
So it really does seem like erythropoietin is acting like some kind of keep-out signal for the immune system. What's the mechanism? How does it actually do that?
They dug into that mechanism. What they found was that in those cold tumors, the erythropoietin being churned out by the cancer cells, it binds to specific receptors, but not on the T cells directly.
Oh, on what then?
On other immune cells called macrophages. These are usually versatile immune cells, can play different roles.
Okay, macrophages.
But when erythropoietin binds to receptors on these macrophages within the tumor, it causes them to flip. They switch into an immunosuppressive mode.
So instead of helping fight the cancer?
They actively start suppressing the cancer-killing T cells. They inhibit them, and they seem to physically push them away, preventing them from even getting close to the tumor cells.
That is quite a sinister tactic by the tumor, using this protein to basically corrupt other immune cells to act as bodyguards.
It's a really clever, albeit detrimental, mechanism of evasion.
So logically, the next step would be to try and interfere with that signal, right?
Yeah.
Did they do that?
They did. They tried disrupting that whole erythropoietin signaling pathway in a couple of key ways. One way was just generally reducing the overall levels of erythropoietin. The other, more targeted way, was preventing the macrophages specifically from being able to respond to it.
How do you do that?
They engineered the mice so that their macrophages couldn't produce the receptor for erythropoietin. So even if erythropoietin was around, the macrophages just couldn't hear the signal.
Clever. Okay, and did that change how the tumors responded, especially when combined with the immunotherapy?
It made a huge difference. This was perhaps the most significant finding. When they combined blocking the erythropoietin signaling, either reducing erythropoietin or blocking the macrophage receptor with the anti-programmed death 1 therapy,
Yeah.
they saw complete tumor regression. The tumors vanished in the vast majority of mice that had those initially cold-resistant liver tumors. And these mice survived much, much longer.
Compared to just the immunotherapy alone.
Oh, drastically different. The control groups and the mice that only got the anti-programmed death 1 therapy without the erythropoietin blockade, they showed very poor survival.
Wow. Any specific numbers on that survival?
Yes, they reported some striking figures. For instance, in the group where the macrophages couldn't make the erythropoietin receptor, just blocking that aspect, 40% of the mice survived until the experiment ended at 18 weeks.
Okay, 40% is pretty good compared to poor survival.
But then, when they added the anti-programmed Death-1 therapy on top of that blocked receptor, 100% of the mice survived. Complete survival in that group.
100%. That's really compelling, even in mouse models.
So let's talk implications. What could this mean for actually treating cancer in people?
Well, the implications are potentially enormous. I mean, this discovery points to a really fundamental mechanism of immune suppression that might be happening in many cancers.
Right.
And understanding that mechanism immediately opens up ideas for new therapeutic strategies.
Like what? What are the approaches?
Broadly, there seem to be two main paths forward based on this. One option is to try and non-specifically reduce erythropoietin levels throughout the body.
But wouldn't that cause problems? Like anemia. That's the major potential downside, exactly. Since erythropoietin is so vital for red blood cells, reducing it systemically could definitely lead to anemia, which would need managing.
Okay, so what's the other approach?
The other, maybe more elegant approach would be to selectively block the erythropoietin receptors, but only on the macrophages within the tumor microenvironment.
Ah, so target the effect right where it's causing the immune suppression without hitting red blood cell production systemically.
That would be the ideal scenario, yes. A more targeted strike.
And this study was focused on liver cancer in mice. Is there reason to believe this isn't just a weird quirk of mouse liver cancer? Could it apply more broadly?
There are actually pretty strong signs that the researchers didn't stop with the mice. They looked at large human cancer databases. They found correlations. Higher levels of erythropoietin, or its receptor, seem to be linked with poorer survival outcomes in people with several other types of cancer. We're talking kidney cancer, breast cancer, colon cancer, even some skin cancers.
So the same pattern seems to show up across different human cancers.
It suggests this immunosuppressive role of erythropoietin might be a much more general mechanism than just in liver cancer. Of course, more research is needed to confirm that in each type.
But the big hope here, the real potential game changer, is that by targeting this erythropoietin pathway, you could maybe take these cold tumors, the ones resistant to current immunotherapy, and turn them hot. Make them susceptible, make them visible to the immune system, maybe especially when combined with existing drugs like the anti-programmed Death-1 therapies.
That's precisely the hope. It offers a potential strategy to overcome one of the biggest hurdles in immunotherapy right now, resistance, making the untreatable potentially treatable.
It sounds incredibly promising. But we should probably also touch on the limitations, right? This is still early stage.
Absolutely crucial. We have to be realistic. First and foremost, this was a study in mice.
Right. Mice aren't people.
Exactly. While these models were sophisticated, what works in mice doesn't always translate directly to humans. We absolutely need further research to confirm these findings in people, and critically, to assess the safety and the actual effectiveness of any therapies targeting erythropoietin signaling in patients.
That's always the big step, the clinical trials.
Yeah.
And even if it does work in humans, cancer is so varied.
That's the other key point, generalizability. While those database correlations are encouraging, suggesting it might be relevant in kidney, breast, colon cancers,
Yeah.
it's almost certain that not every single tumor, even within those types, will respond the same way. Cancer is incredibly complex, tumors are heterogeneous, so extensive research across many different human cancer types and subtypes will be absolutely essential to figure out which patients might benefit most from this kind of approach.
Definitely. It's rarely a one-size-fits-all solution in cancer. Now, the senior author on this paper, Dr. Edgar Engelman at Stanford, he seemed quite excited about this work. What was his take?
He was very optimistic. He described the findings as a fundamental breakthrough in just understanding how the immune system is regulated within the context of cancer.
A fundamental breakthrough. Wow.
Yeah, strong words.
Yeah.
And he expressed real hope, optimism, that treatments based on this discovery, targeting this mechanism, could move into human clinical trials relatively quickly.
That would be fast.
It suggests they see a clear path forward. He also really highlighted how surprising this all was, pointing out that because erythropoietin is so well known for its role with red blood cells.
Yeah, it was kind of hiding in plain sight.
Exactly. It apparently took quite a bit of digging and careful investigation to finally connect the dots between erythropoietin and immune suppression within the tumor itself. It wasn't an obvious link to make initially.
It really underscores that idea, doesn't it? How sometimes looking at biological processes we think we understand completely can suddenly unlock these totally unexpected insights into really complex diseases like cancer. So as we kind of wrap up this deep dive, what's the main takeaway or maybe a provocative thought you'd leave our listeners with?
I think for me, this research is just such a powerful reminder of why basic science, fundamental investigation, is so incredibly important. By really digging into the details of fundamental biological mechanisms, even ones we think are, solved or well understood, like erythropoietin and blood,
Yeah.
we can stumble upon these surprising connections that open up entirely new and potentially transformative ways to tackle really challenging diseases like cancer.
So don't stop asking basic questions.
Never stop. It really encourages us to keep that broad perspective, to appreciate just how intricate and interconnected everything in the body truly is. Unexpected discoveries are often waiting in places we thought we already knew completely.
That's a fantastic point. And for listeners who are really interested now and want to follow this specific story, what should they keep an eye out for?
Well, based on Dr. Engelman's comments, the next big step would be clinical trials. So keep an eye out for news about early phase trials, phase 1 trials probably, investigating therapies designed to target erythropoietin signaling in cancer patients.
Looking for drugs that block erythropoietin or its receptor on macrophages.
Exactly, particularly in combination with existing immunotherapies like the anti-programmed Death-1 drugs. As he suggested, there's hope these might start appearing relatively soon. It's definitely a space to watch. This research feels like a very significant and genuinely encouraging step forward in tackling those really tough-to-treat cancers.
Definitely something to follow. Thanks so much for breaking that down for us.
My pleasure. Fascinating stuff.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.